Welcome to our GCP-Mindset channel. In today's web seminar, we talk about sample size calculation in clinical trials.
I would just basically outline the importance of the topic. I think sample size calculation is what we're going to talk about today, and it's a very important topic. And it's a topic we understand is not fully comprehended by everyone that is working in the clinical research field. We have a lot of clients of varying degrees of experience coming to us, asking for our support, and the justification of sample sizes within the proposals of the studies that we are trying to support vary quite a lot between studies, from not having a justification at all to estimations or references to other studies that were conducted in the past to actual sample size calculations and justifications. And so today we would like to use the space to talk a little bit about why we needed sample size justification to begin with. And based on that foundation, we want to talk a little bit about statistical principles behind sampling and hypothesis testing. This will not be a statistical webinar, so if you're joining for the in-depth statistical knowledge, I'm afraid that will not be the focus of today's webinar, although I are always happy to discuss it also after the fact. Today should be more about the grand scheme of things to help enrich the knowledge of this topic in the wider group of people engaging in clinical research. So there will be a little bit of a dip into the statistical side of things, but not too heavy. And a strong focus of this presentation will also be how sample size justification and determination differs between different study phases, which can also, of course, then be applied to different study concepts. For example, medical devices are, of course, different and don't have study phases. And lastly, sort of as a public service announcement, we want to briefly highlight difficulties or dangers when using online calculators for your sample size justification, which we know are commonly used, especially amongst those clients of us that don't have a dedicated statistician that is supporting them in these questions. So why do we need a sample size justification? Well, first and foremost, it's a problem, an ethical problem. I think this is very important to understand because you will need to justify sample sizes within clinical trials, also in preclinical trials, and you will need to justify it also in front of ethics committees. And in ethics committees, you will always also find a statistician on board that very much so looks at your study design, at your objectives, at how you want to analyze your endpoints, and whether all of this aligns with the sample size you have suggested. Why is this? Well, because we have a clinical trial that is basically using a potentially unsafe drug or medical device. We are trying to gather information to basically enrich our understanding, and we don't yet know about the safety profile of our drug or our devices. Of course, the later on in the development phases, the more we know. And if something similar is already available, then of course we also have additional knowledge. But in general, we are always running a certain risk for patients, which may differ between trials. Also, more efficacious treatments may be available. And therefore, really, you need to make sure that the data you generate is meaningful enough to justify these issues that potential participants in your clinical trials could encounter. So when you look at a clinical trial, it is not necessary that the participant itself has an advantage. In most cases, that's not necessarily given. It's quite clear for phase 1 studies, if you use healthy volunteers, that they do not benefit from receiving a certain drug, for example. Once you go in the indication, you may have placebo-controlled clinical trials in which, of course, the placebo group does not have an advantage of participating in your study. And what you need to focus on is how your study, and in line with your study, also your sample size, helps further scientific knowledge and helps advance society overall, and not only your patient group. And so by keeping sort of like patient burden within a clinical trial limited, but still gathering enough data to support further development and to support helping patients in the future, that's what the sample size is basically doing for you in your clinical trials. And when it comes to the sample size, what you don't always need to do is calculate it. And we will go through the different study phases also, and you will see that for a lot of the trials, there is not really a means to calculate your sample size. But what you always will need to be doing is justifying it. Justifying the size of the sample that you want to include in your clinical trial, and that exposing the final number of patients that you have decided upon in your clinical trial is justified when weighed against the benefits that society could have and the risks that you're exposing patients to. And the core here is to always go as low as possible, but as high as necessary to show enough robust data to come to a valid conclusion that helps you move on into the next phases of your study. And with that, I will basically hand over to my colleague Matthes, now that you've understood sort of like what we're talking about here, to discuss sampling and moving into statistical tests.
Yes, thank you, Andy, and thank you also to having me here. And yeah, and thank you for taking over the introduction. And welcome from my side, and I will start with the general concept of statistical hypothesis testing. And each project starts always with the research question. And the research question is regarding a target population. That could be the whole population of the Earth. It could also be a subgroup of it. But in most cases, and I think in nearly all cases, it's not possible to really investigate the complete target population to answer your research question. And therefore, we have to draw a sample from our target population. And with that sample, we are trying to answer the research question. And how we do this? We don't do this with magic, but we do this with a statistical test. So the idea is that we have a research question, then we will take a sample of our target population, and with that sample, we try to answer the research question, and we conclude on the target population. So to give this a little bit more content, we have an example that the research question is, for example, that the average treatment effect is higher under verum compared to placebo. And in this phase, there is a discussion ongoing, and a lot of discussion is necessary here, also with some stakeholders. What is really the research question of interest here? But when we have completed this phase, we will set up some statistical hypothesis. And this can you see here? And the idea of a statistical hypothesis is that we have all possible outcomes here, the treatment effect denoted as delta, and we divide all possible outcomes into two areas. We have the alternative hypothesis. This is what we want to show. In our case, that we have a treatment effect which is beneficial for the verum group. And on the other side, we have the null hypothesis. This is what we don't want to show. In our case, that there is no treatment effect, or that even placebo is better than the verum group. And all the things here are done prior to that we conduct the study. And when we have set up the statistical analysis, then we start to collect the data. And with that data, we make our statistical test. And the idea behind the statistical test is that we test the null hypothesis. And we want to show that based on the data we have collected, it is very unlikely that the null hypothesis is true. And as the null hypothesis is not true, the alternative must be true. Therefore, the idea is the null hypothesis could be rejected, and therefore the alternative should be accepted. And this is, as the alternative is, this is what we want to show. The next two slides are a little bit more technical, and we go into more statistical details, but I promise this will be the only ones in this webinar. So when we have the general scenario we have is that we have a test decision, that our test could say that he prefers the null hypothesis, or the test could say that the alternative is true. This is what we have here in our columns. And what we have in the rows here in this table is the reality, what is in reality the case. For example, if we look at this row, we would say that in this case, the null hypothesis is true, and this means that there is no treatment effect or a better treatment effect for placebo. And when we look at this row, we would have that we have in reality really a beneficial treatment effect for the patient. And what we now can do is, if the in reality the null hypothesis is true, meaning that there is no treatment effect, and the test said that is also no treatment effect, then this is a correct decision. But nothing is gained from here, because we just identified that our drug has no effect. But what we really want to show is that if in reality the alternative hypothesis is true, meaning that there is a treatment effect, and our test also said that there is a treatment effect, meaning we are in this column of H1, then this is also a correct decision. And what we have here is we have found a treatment effect. We have correctly found a positive treatment effect. But as we do some decisions, we can do some errors. And there is the so-called type 1 error, which is the more important error. And the type 1 error is that in reality, there is no treatment effect, but the test said that there is a treatment effect. And in this situation, we would have a drug on the market which is not effective, because it's in reality, there is no effect, but the test said there is an effect. And this type of error is more important, all statistical tests are designed so that the type 1 error is controlled. There could also be a second type of test, and this is the so-called type 2 error. And this is when in reality there is a treatment effect, a positive treatment effect, but the test said we should have accept the null hypothesis, and therefore this is a wrong decision. And that means that although there is a positive treatment effect, the treatment will not come to the market. To illustrate this a little bit, we have a graphical illustration of it. And when we remember that if it were possible that we could test the complete target situation, we would have the following situation. We would have here on the x-axis would have the treatment effect, and if we would investigated the whole target population, we could decide after the investigation whether the null hypothesis is true or the alternative is true. But as I said, we have to draw a sample, and therefore we have to make a decision under uncertainty. And what we see here is we have on the x-axis the treatment effect, and we have here the distribution under the null hypothesis. That means if we would conduct the study more than once, we would draw from this distribution. That means in most cases we would get a treatment effect which is close to 0. In some cases we would observe that the placebo is very good, and in sometimes we would, although we are in the null hypothesis, meaning that there is no treatment effect, we would observe a positive treatment effect. And what the statistical test does is to set a threshold, and we would then conduct the study, and if we would observe a treatment effect which is higher than the critical value, we would conclude that the null hypothesis is too unlikely to be true, and therefore the alternative is true. But as here highlighted in the dark blue area, that it is possible that although the null hypothesis is true, we would our treatment effect would be higher than our critical value. And this dark blue area reflects the type 1 error that could be done. On the other hand, when we have treatment effect from the alternative, in this case it is close to 4, And also here, when we conduct the study several times, in most cases, we will get a treatment effect around 4, sometimes a little bit higher, but it is the possibility that we would observe a treatment effect lower than our critical value. And this is in the dark red zone here. And if we would observe a treatment effect from the dark red zone lower than the critical value, we would not reject the null hypothesis, because we would say that it is in accordance with our null hypothesis. But in reality, the treatment effect would come from the alternative. And therefore, this dark red area here would represents the type 2 error. And what we have left is the light red area. And this light red area reflects the power in this scenario. And the aim of a statistical test is to maximize or to optimize the power in a statistical test. Because, to just a short reminder, that the power is the probability that we correctly reject the null hypothesis and conclude that the alternative is true. So in the next slide, we see that there are a lot of factors that have influence on the power. There is the significance level, there is the type 2 error, there is the difference between the groups that we want to show. Also, the variance in the data has an impact on the power and the sample size. And we will go through all these factors and see how it's influenced the power. We start with the significance level, the maximum type 1 error. And what we see here is in this, to remind, is that the light red area we want to maximize. And when we increase the significance level, then the critical value decreases. As you can see that the area, the blue area here, will increase, and the red area will also increase. So we see that when we shift the critical value towards 0, the power is will be higher. To say this in words, the higher the maximum type 1 error, the higher the power will be. Or we can also put it the other way around to say when we have a higher maximum type 1 error needs a smaller sample size. When we look at the treatment effect, this is the shift of the true distribution, and we see the higher the treatment effect, the more the alternative is shifted toward infinity, and then we see that the light red area increases. So the higher the treatment effect, the higher the power. Or we can also put it the other way around. When you have a higher treatment effect, then you need less patients. The variance in the data has also an impact on the power, and we will see when we have more variance in the data, meaning that we have here that the distribution are getting wider, then we see that the power is also decrease. So we can say the smaller the variance, the higher the power. That means that you have a higher precision in your estimation. And you can also say it regarding sample size, the smaller the variance, the smaller is the required sample size. So to sum up, with the factors, we see that the significance level has an impact on the power, but there's a common threshold. In most cases, the significance level is of 5%. Sometimes it's also 2.5%, but in most cases, it's 5%. So this is set, and the type 2 error is also in the most cases set. It should be at least 20%. Sometimes it's also 10%, but it is also a fixed number. With the type 2 error fixed, we also have the power fixed, because this is 1 minus the type 2 error. And then we have the difference between the groups that should be shown, and this is nothing we can change. This is just the reality, and we want an estimation, or we must assume a treatment effect which is really realistic. When we overestimate our treatment effect, then we will not be able to show a lower treatment effect. So this is a number we must give a good guess so that we achieve the correct power. The same is true for the variance. This is nothing we can change. This is a value which is fixed in our model, and therefore we have no possibility to adapt something in the difference and in the variance. That means that the only factor left is the sample size. And that is the reason why the sample size is so important in clinical trials, because this is the only factor we can change so that we can achieve the desired power. Then there are two settings in which statistical studies could be conducted. And we have the confirmatory study, where we do the statistical hypothesis testing. Therefore, in this scenario, we want to conclude on the target population. And therefore, it's very important that the type 1 error is controlled. And we call this the inferential statistics. And in a confirmatory trial, we would say something like that treatment A is significantly decreased the recovery time compared to placebo. Just the result of the statistical test and nothing more. But the advantage is that we can conclude in a confirmatory setting on the target population. On the other hand, we have the exploratory approach. And in an exploratory approach, we will do a lot of statistical tests. We will produce some p-values and so on, and test different subgroups and different endpoints and so on. And it's okay, because the type 1 error is not really of interest here. It's just a description, just a descriptive statistic. We want to describe the data we have observed. An example would be that we really just say what we have observed, but we do not really conclude on the target population. This will be done in a further trial, in a confirmatory trial. And another point that arises is that when we test several times, then the type 1 error is not controlled anymore. And there is this very interesting website who have a very big data pool, and they looked into the data and tried to find some spurious correlation, how they call it. And we have here two examples. For the first one is that the US crude oil imports from Norway correlates significantly with drivers who are killed in a collision with a railway train. And you see, this is a perfect correlation over the years, but nobody would say that this is really a causal relationship. The other example is that the number of people who drown by falling into the pool correlates very good with film Nicolas Cage appeared in. And this is also, but the correlation we observed here is just an observation. And this is the reason for it is that we have make a lot of tests so that we have this so-called multiple comparison problem. If we test a lot of times, then we will find something that will correlate, although nobody would, in this case, it's clear that this is not really a causal relationship. But when you have a clinical trial and you are expect that there is something, yeah, might be more prone to believe that what you see. And to give also an impression how the type 1 error is inflated, we have here on the x-axis the number of tests we conduct, and we have on the y-axis the maximum type 1 error. And we see when we have one test, then the type 1 error, the maximum type 1 error is controlled to our significance level of 5%. But when we test twice, then we see that the type 1 error is already inflated. And when we see, we have a table here where the exact values are displayed, and we see when we test 14 times, then the maximum type error is over 50%. And of course, this is always, there's the possibility to adjust for multiple testing. We have here an example where we have the simples adjustment, the Bonferroni adjustment. But we see here on the green line that when we correct for 2 tests, then when we do 2 tests, then the type 1 error is controlled. But then it's also get inflated. And the bottom line here is that there is possibilities and methods to adjust the type 1 error, but there is no method that you can test as much as you want.
Thank you for watching part 1. In the next part, we talk about the role of sample size calculations throughout all study phases. See you there.